Hi, welcome back to 131. This is chapter 28 on eukaryotes, part 3. And so I've done the first one on what is a eukaryote. The second part was long, and it was on how eukaryotes came to be from archaea and the origin of the nucleus endomembrane system, the mitochondria, and the chloroplast. And so I'm going to take up now talking more about the diversity of the different types of eukaryotes. And they're broken up into four what we call clades. We went over this in lab, that on systematics. But a clade, if you remember, is just any branch point in a phylogenetic tree. And these clades will be grouping together kingdoms into the domain. So we have one big domain, the eukaryotes. Then we have separate kingdoms, three of which you probably already know about: the animals, the fungi, and the plants. And then we're going to see there's a whole bunch of other kingdoms, and some of these kingdoms are grouping together. And so they're not grouping together enough to make a domain. These are within the domain. So whether we want to call them super kingdoms, subdomain, we're just going to call them supergroups. That's what your textbook does. So looking at that, we have to look at their origins a little bit. And one interesting thing is that in one group it's very clear, the Archaeplastida, they have the chloroplast that we talked about last time that came from engulfing the cyanobacteria. But in these other groups, we have different groups that have some members have chloroplasts and other members don't. So we have to explain that. And that's going to be our step 4 in my diagram. And this is going to be secondary endosymbiosis. And so you probably already can guess by the term endosymbiosis how it's going to come about. And then I wanted to finish up by talking about some things that if you've had 130 or 102 recently, you can kind of cut out at the end. And that is the importance for life cycles of the terms. We'll talk about ploidy levels. They're just starting right now with haploid and diploid. There'll be others later on in the semester. And talking about mitosis and meiosis. And so let's get going. And so we're now in our third part, talking about serial endosymbiosis and other chloroplasts and the outcomes of mitosis and meiosis. And looking at our diversity, the eukaryotes are divided up into 4 supergroups within each one of these groups. And so if you were to look down here, okay, this is showing you eukaryotes, and you know that you're a eukaryote. Where would you be? All right, you're down here in the Uniconta. And that's showing you an amoeba that you might recognize from lab. And that's one of the groups. The amoeba zones and the Phystoconts are 2 groups of kingdoms that make it up. So both you and a fungus are in the Uniconta. So the Uniconta comprises more than one kingdom, because we know the animals make a kingdom. The Archaeplastida are the plants and red algae, and you know plants are a kingdom. Then we have 2 other groups you probably can't relate to without having studied lab yet, unless you've already learned something about protists and plankton and things like that. That'll be a clade that's called the SAR. It's 3 different groups, the first letter of each one, and the excavata. And so we have the excavata, SAR clade, Archaeplastida, which I've talked about before, in the Uniconta. And so these are clades within the group. And so if we look at these major groups, and so looking at Archaeplastida, we have plants, that's a kingdom. We have the green algae, they're considered a kingdom, and the red algae. So these are groups of kingdoms coming together, but they're within the same domain. And so it's something between the kingdom level and domain level. And so in lab, we're going to see, I'm trying to and I've highlighted in yellow so you can see, there you are down here, showing you at the high level that we're talking about, that you're just one little thing in with the quinoflagellates, fungi, animals, amoebas, and slime molds. That's in our group, the Uniconta, which is in the Phystoconts within the Uniconta. Okay, and we'll get to that later on. So anyway, in lab, you'll be seeing a diversity of these organisms. I can't show you all the diversity. And so what I've done is I've chosen one organism from a few of the clades. So for example, if I were to show you a kangaroo and say that's an animal, all right, so you're going to see one. And so some of these, we'll do plants in a little bit more detail, and we'll do the fungi in a little bit more detail. But you may recognize some of these already from lab. Here we have a foraminifera, we have a chlamydomonas here, we have euglena. So these are some of the organisms that we'll see in lab. Okay, so we have the 4 clades, the 4 supergroups, right? In lab, we're going to be going over the diversity, but why they are broken up and grouped together and separated, I guess is a better way of saying it, is based on their nuclear DNA. And I use the term nuclear there because we're going to contrast it with chloroplast and mitochondria. And so the mitochondrial DNA is also useful in this group because the mitochondria had one origin and is followed through these different lineages. So nuclear mitochondria DNA is a good source to show how these things relate. But chloroplast DNA actually tells a little bit different story. That's what we're going to get into when we get to serial endosymbiosis. And so that's because some of these groups have chloroplast. Obviously, the archaeplastida, the red and the green algae, all have chloroplasts, right? With very few exceptions of some of these parasitic plants. But then we have other groups where many of the members have chloroplasts and some don't. And that's what we're trying to figure out. So we see this, and so how did that happen? So if this is looking at our diversity of the clades, so these would be our four groups. They're labeled over here. So this would be the excavata, the SAR clade, the archaeplastida, the uniconta, and then what they're showing you in yellow are the organism that we call protists. And you'll notice that it doesn't have a single common ancestor. It's really just describing everything in the eukaryotes that are typically not or have members that aren't multicellular. And so it pulls out the three major multicellular kingdoms, the plants, the fungi, and the animals, and refers to all the others. Kind of breaks down because some of the red algae are quite big, and up here the brown algae, those are the kelps, those can also be quite large as well. But most of the other ones are all single cells or very small multicellular organisms. So if we look at this clade, and I'd say, well, where are the chloroplasts? Initially, without knowing much, you'd say, well, archaeplastida, we know from when I said, one step 1, 2, and 3, that was our step 3. And so that chloroplast got there and gave rise to the chloroplasts in the red and the green algae and the plants. They all have chloroplasts. But if you notice up here, there's at least one word should pique your interest. The brown algae, they have a chloroplast. Dinoflagellates have a chloroplast. Diatoms have a chloroplast. Some euglenas, most euglenas have chloroplasts. And so if we look, but the oomycetes in that clade don't. The apicomplexans and the cilia, so we know it's not that they had a chloroplast and it followed all the way up. And so they seem to have acquired it after the group has separated out from the others and evolved. And so we have our origin of our chloroplast. That was our step 3 in my 1, 2, 3, 4, and we're going to go over step 4 in a minute. So that's the origin of the chloroplast. But then if we look at these other groups, as I just mentioned, they all have chloroplasts as well. And so we have to figure out, how did the brown algae get the chloroplast? The diatoms, dinoflagellates, and euglena. These are the ones we'll be seeing in lab. There are others, but these are the ones we'll focus on because we'll see a number or two of them in lab. Around here, they're all, euglena is not significant, but the brown algae, diatoms, and dinoflagellates are very significant for us along our coast because those are very important in the aquatic food chain in the ocean near our shores, along with cyanobacteria. So you may hear quite a bit about them, especially in relations to climate change, which we'll talk about towards the end of this lecture. And so if we look, we have the origin of the chloroplast here. It passes on from daughter cells to daughter cells. So that was our step 3. How did these ones get it? And what they did is they got it by eating either a red or a green algae. And so the red algae became the chloroplasts in these 3 groups that have them brown because they actually have kind of a brown color. And then a green algae became the chloroplast in the euglena. And this is our step 4 that we're going to talk about. And so from the green algae, it became a chloroplast in the euglena. Right? The diatoms, brown algae, and dinoflagellates also incorporated by endosymbiosis, a very similar story than we heard about before for both the mitochondria and then the cyanobacteria, and now for chloroplasts eating other algae, or other algae being eaten, turning into chloroplasts. And so this would be our step 4, the other chloroplasts. We're going to call this secondary endosymbiosis because the first one in this case was the original cyanobacteria, so that's the first. in terms of the origin of the chloroplast. And that was my step 3 before. So when I was going over step 3, this is what we're talking about. That was step 3. And so it became an algae, and this would algae actually would be either a red or a green. It's in the Archaeplastida. Obviously, the green algae gave rise to plants, but they're too big and they're not involved in the story. They come later, actually. So the green algae come first, plants come later. And so we have these single cell red and green algae. And then another, let's say a Stromanopila, comes along, and notice it doesn't have a chloroplast. It's like amoeba. It eats the red algae, and as we saw before, it survives as an endosymbiont inside the Stromanopila, and slowly over time, so notice it has its own nucleus, its own mitochondria, and its own chloroplast. And when it's first eaten, it maintains all those, but over time, the algae loses its nucleus, loses its chloroplast, loses most of its cytoplasm, and just retains what was its chloroplast, now is a chloroplast in the new algae cell, and in this case would be a brown algae. So it's a very similar story to what we saw with the origin of the chloroplast. Okay, so this is our step 4. So the first 3 I went over, I'll briefly review. All right, so we went over how we had the formation of the proto-eukaryote cell by invagination around the nuclear material, separating off these specialized membranes as pockets, becoming the nucleus and endomembrane system. Right now we have a prokaryote cell. Somewhere along the way it got a cytoplasm and cytoskeleton and can do phagocytosis. Second step is this cell, now that it can do phagocytosis, this proto-eukaryote engulfs the aerobic bacterium, and that turns into the mitochondria. Some continue, so those other 3 clades we're talking about, the excavata, the uniconta, R clade, and the SAR clade, just derive off step 2. Right? The Archaeplastida, one of these groups that evolved, had a mitochondria. It took in the cyanobacteria, and that became the chloroplast in the Archaeplastida. That's our step 3. Now what we have in step 4 is some of these descendants from over here, so someone in the Stromanopila and excavata, they took in an algae, and that became their chloroplast. So that's our step 4. We call it secondary endosymbiosis, right? So notice, first, this one is very different in that it's all eukaryote cells. Everybody's eukaryote by now. It's a heterotrophic, meaning eating others, doesn't have a nucleus. Something I would say like an amoeba, but the uniconta actually didn't do this, but something of that kind of form, a euglena ancestor that didn't have a chloroplast, then eats a green algae. Green algae are already eukaryotes. So this is all eukaryotes at this stage, right? And so whether it's a red algae or a green algae, determines the different groups and type of chloroplast they're going to have. So they're engulfed by different heterotrophic eukaryotes. Those are either in the excavata or the SAR clade. And over time, we get that same simplification process, losing the cell wall, losing their nucleus, mitochondria, most of the cytoplasm, but retaining the area around the chloroplast. And we have our 3 groups. And so the Phaeophyta, that's the brown algae. The diatoms and dinoflagellates are derived off the red algae, and that's why they have a different color. They're not bright green. And the euglena and the cercozoans, which I'm not going to talk about, we'll see euglena in lab, or I'm not going to talk much more about the cercozoans. I'll just give you one example. Those are green algae, and that's why euglena looked so bright green in lab, just like Elodea leaf looked, because they have that same type of chloroplast. Okay, reviewing from last time, step 1 was the formation by invagination of the eukaryote cell from an ancestral prokaryote that we know to be an archaea cell. Then step 2, you engulf the heterotrophic aerobic bacterium that becomes the mitochondria. These give rise over here to the other clades, the Uniconta, the Sarclade, and the Excavata. Then in the Archaeplastida, which already has a mitochondria, it took in the cyanobacteria and became the Archaeplastida, the red and the green algae. And so I've added the step 3 and 4 here because the organisms, the way they're drawn in the book, this step 3, this organism, is that organism. They look really different, but it's that same process. So this organism is this process here. They just drew flagella on it. All right, it's this organism taking in a cyanobacteria. That's our step 3. Here it is again. What they're showing you here is the membrane layers, because this is what gives away the story. And so the green algae is the endosymbiont this time. And so when they draw the lines, the line really should go to there and to there. There's your green algae. Here's your red algae. It's been eaten, right? So in here, this is a euglena ancestor that doesn't have a chloroplast. It takes in the green algae, and for a while it has a green algae growing inside the euglena, and then it gets simplified down into being a chloroplast in the euglena. And this is the chlorachnophyte. I'll talk a little bit because it has this little vestigial nucleus. Notice there's a little purple dot that doesn't go away. We'll talk about that later. Same thing with the red algae, and this is happening in 3 groups in the Straminopelas. It's happening in the it's happening in red algae, the brown algae, diatoms, and dinoflagellates. Easy way of saying it. And so you can see the same thing. It's being taken in by, let's say, a dinoflagellate ancestor. It survives as an endosymbiont for a while, and then it gets simplified in simply being a chloroplast in either a dinoflagellate or a diatom, which they're not showing you here, or in a brown algae, which they're showing you here. So again, seeing the steps going through, taken in by endosymbiosis, and then simplified. Taken in by endosymbiosis, and then simplified. Okay, so I want to give some evidence of this. And one of the more interesting things is a couple of things. One is the number of membranes, and that's what they're showing you here. And so if we look and count, remember when we gave evidence for both the mitochondria and the chloroplast, when it was taken in by phagocytosis, it got that outer membrane, which was eukaryotic, but it maintained its inner membrane, which was prokaryotic. So in both the mitochondria and the chloroplast, we have an inner bacterial membrane that actually does all the work in either of those cases. The one with the electron transport chain, if you've had 130, that's the prokaryote, that's the bacterial membrane. Right? The outer surrounding membrane is eukaryotic like the rest of the cell. As you would expect, it was taken in by phagocytosis and became a food vacuole. Here, the interesting thing is, if you look at the membranes of the chloroplasts in the dinoflagellates, diatoms, brown algae, euglenas, right, chloranophytes, they have an extra additional membrane to the outside, and that's the remnant from being taken in by endosymbiosis. And there is some more evidence than that, but it's just, this is just counting those membrane layers, and that's what it's showing you. Other evidence was kind of interesting in one of the groups, and you don't need to memorize this. If you notice, there's something different about this. So here we have our nucleus in the chlorachnophyte ancestor or the euglena ancestor. And of course the green algae, being a green algae, eukaryote, has its own nucleus. But over time, you'll notice that in the euglena, in the chloroplast, it disappears. As happened in the dinoflagellates and in the brown algae. So here we have 2 nuclei. There's the nucleus from the ancestor cell. The red algae has a nucleus. But as it gets over time, the nucleus is only in the cell, and there is no nucleus in the chloroplast. It's become simplified. But there's one group that's kind of interesting. You notice right here in the chlorachnophytes, chloranacophytes, it's hard to say that. You can see it still has a vestigial nucleus. They call it a nucleomorph. And so it is still dividing inside the cell. So you have a cell that divides by mitosis, and this divides also by mitosis. So its chloroplast divides by mitosis. Inside the chloroplast, its chloroplast membranes divided by binary fission. It gets a little complicated there. But anyway, so this is just showing you some more evidence of remnants where it hasn't completely simplified. Right? So not fully simplified is what's happening here. And so that gives us some evidence of what has happened in the other ones, where they've completely simplified and lost it, because this gives one that still has it there. Referring to the nucleus in that case. Other things about the arachnophytes, it's kind of interesting. Don't need to memorize them. They have this vestigial nucleus called the nuclear morph. There's a whole other group, the Paulina, that actually have peptidoglycan cell wall remaining between the membranes of the chloroplast. And so it hasn't fully lost its cell wall from when it was a cyanobacteria. And it still uses phycobilisomes, which I talk about in lab, to capture light. And so its chloroplast is still very much more similar to a cyanobacteria than other ones. And this probably has come about as being a separate endosymbiotic event, separate from the rest of the Archaeplastida. So that's kind of interesting as well. Right? Just more evidence. And so do not memorize this, but this is just showing you, and if you get into cell biology, there's a lot more complexity than what we're going into. But it's just showing you that you can actually have, here's what I was just showing you for Paulina. Notice it's got its own origin of a cyanobacteria separate from all of the other algae that we have. And so apparently this has happened possibly twice in all of history. And this is showing you the arachnophytes that still have their vestigial nucleus here. And it's just showing you all the different stages and how they study this. And so if you wanted to get into the origin, it's a bit more complicated than we're simplifying, but there's lots of evidence to support this. And this is a little tricky because if we're looking at our clades, this is secondary endosymbiosis, but how we're relating everything is with, see if I can get this, here we go, the nuclear DNA. And so secondary endosymbiosis is bringing things up from the red algae. And so at some level, if you're looking at it, you would say the diatoms, brown algae, and dinoflagellates share structures with the red algae. You might think they're more closely related, but you can see they're very different clades in the groups. They're not that closely related. Same thing if you're looking at the euglena, it's bright green chloroplast, and we'll see euglena in lab. You might think it's more closely related to the green algae, but it's in its own clade way off on the excavata. And so something to keep in mind when we're looking at saying how things relate. Okay, so that's our serial endosymbiosis. That's our step 4, which explains, let's see if I can find my step 4. There we go. So we have 4 different steps. This is the one I just went over, and this is explaining the unusual chloroplasts in the other clades other than the Archaeplastida in the eukaryotes. Okay, now I'd like to switch and talk a little bit about something that all eukaryotes have in common and having to do with a little bit of cell biology and some terms, and it has to do with reproduction and life cycles. And so before we do that, just want to mention, because we're going to be talking about mitosis and meiosis, and in this process, the chromosomes are pulled apart. In any movement within the cell, and I mentioned it earlier when we were talking about phagocytosis, you need a cytoskeleton. And that same cytoskeleton was what makes up those microtubules that I talked about, giving you the 9+2 in the flagella. And where that came from is still kind of a mystery. And so that's something in that stage 1 when the cell was becoming eukaryote from the archaea ancestor. Where we get to mitosis and meiosis may seem like a big jump, but it's not quite. It's basically just a variation off of binary fission. But being haploid and diploid is kind of a consequence from that that's important for our life cycles in the eukaryotes, which are very different from the prokaryotes. So we have a life cycle is what we call it in eukaryotes. And in eukaryotes, we have 2 ways that cells divide. And that is mitosis and meiosis. And in mitosis, we have a cell, and I'll just draw 4 chromosomes. It will divide into 2 cells. with 4 chromosomes. We have another cell division called meiosis. My pen is, start over there. Come on, pen. Pen has stopped working. Okay, we'll go back to our we have 2 types of cell division. We have mitosis and meiosis. I'm trying to recharge my pen while we do this. And what they do is they divide. So in mitosis, you get exact copies of the daughter cells. In meiosis, you get a change in ploidy level. And I just want to point out there's a diversity. There's a huge group. So if you imagine how different an animal is from a plant, from a fungus, and then from these other ones we're looking at in lab, like a diatom, there's a huge diversity in the way that the cells do this. And so what we get are different types of what we call life cycles. And some form what we call resistant cells, which we really don't have anything like that in animals. Some animals kind of go into a dormant phase, like hibernation. Some of the other ones, like some of the arthropods, actually do make resistant egg structures that can freeze over winter and dry out and come back when there's water, like the shrimp in the ponds in the Central Valley. But by and large, we don't have it. In other ones, you get certain cells called cysts or spores that are very resistant, that are in many eukaryote life cycles as well. And so there are 2 terms I want to go over with the life cycle. First is haploid, and the second is diploid. There are more ploidy levels that we'll get to later in the semester, but we'll get over, talk about them when we get to the fungi life cycle in particular, and then the plant life cycle later on. And so what makes it haploid is that it has 1 copy of each gene, and it's made up basically of 1 complete set of chromosomes. You want to remember that, 1 complete set of chromosomes. In the animal life cycle, it only is found in the egg and the sperm, typically. There's lots of exceptions, like drone bees that are haploid. In plants, they can be in the spores, or you can have actually a whole haploid plant. In the aquatic organisms, you can have both haploid and diploid, but haploid swimming organisms that can be going around and also make a whole plant. And this last thing I put in there is because if you've had 130 or 102, the way they describe biology and cell biology at the introduction level is they make everything either a plant or an animal. And in animals, your haploid cells do not grow and divide. Your sperm and your egg don't divide and go off by themselves. Right? Looking at the equivalent in other life cycles, they would. And so I just want to point out that mitosis can take place in haploid cells. And if you look at the vast majority of all the eukaryotes, it does. Is that animal's unusual in that it doesn't. Diploid is where you have 2 complete sets of chromosomes. So that means you have 2 copies of every gene. And when you do genetics, you'd be looking at, whether you have wavy hair or not, and you get 1 set of genes from your mom, 1 gene copy from your mom, 1 from your dad, and you're the combination of the 2, right, when you study your genetics. And so you have 2 complete sets. I want to notice that that we refer to the set of chromosomes as N. And so haploid is just N, where diploid being 2 would be 2N. We'll get to it later on, but plants have a phase that's called triploid, and that would be 3N. Also, because a lot of people think still, students sometimes get confused from learning about plants and animals only. They think that haploid is only eggs and sperm and they can't do anything. And they somehow think that the haploid isn't complete and functioning. I want you to notice that it's not like the diploid is N and haploid is half N, meaning it's missing something. Haploid is N. It's the whole complete set. Diploid is 2N. It has 2 whole complete sets. And so diploid cells obviously can reproduce. They can reproduce by mitosis. But what diploid cells can do that haploid cells can't do is they can divide into daughter cells that only have half as much chromosomes as they started with and basically turn into haploid cells. And of course, the way you get diploid cells are by two haploid cells, an egg and a sperm, coming together to make the diploid cell. Okay, going back, prokaryotes are only, that's in the wrong spot. Prokaryotes are only haploid. In eukaryotes, there's usually both a diploid and a haploid stage. And the diploid stage is coming about by the fusion of two haploid nuclei called karyogamy or syngamy. Fertilization is another term that's used for that in animals in particular. Mitosis sometimes isn't followed by a division of the cytoplasm. So mitosis is literally talking about the division of the nucleus. And so in lab, we'll see some types of slime mold that actually tend up to these big blobs with lots and lots of nuclei in them. And that's because they're doing perfectly normal mitosis. It's just they're not dividing their cytoplasm. And that results in multinucleate cells. And there's lots of variation in the different types of life cycles. And unlike humans that do not have it, animals, and so by learning life cycles with animals, it's kind of a disservice because it's so simplified that we're missing out what the vast majority of other organisms are doing. And they usually have alternating between a multicellular free-living haploid phase and a multicellular free-living diploid phase. And in animals, we don't typically have that. We have just the diploid organism makes the gametes either fuse or they die, and if they fuse, they make the next diploid organism. We don't have that multicellular haploid phase that we're going to see in a variety of organisms. Okay, so just to show you some complexity, this is malaria, Plasmodium, which we will be looking at in lab, not live, just prepared slides. And we're just going to be looking at this stage here that's infecting the red blood cell. But to understand this, first of all, it's showing you the complexity of life cycle. For this organism to complete its life cycle, it needs two hosts. It needs a mammal, actually a vertebrate, and it needs, it's not showing it here, the gametes, fertilization coming together. It needs the mosquito. There it is. This is the mosquito, and the mosquito is hard to see. Mosquito to work. And so we'll start with what we see. So we have the reproductive cells are forming in your red blood cells. These are gametes. When the mosquito bites you, it sucks up some of those gametes into its stomach. Inside the mosquito, the gametes come together, make the zygote. The zygote erupts into oocyst, and the next person it bites, they're showing it the same person here. So later on in the stage in the life cycle, in the mosquito, it will bite you, and then now it infects you with the oocytes that go to your liver, and then they make the merocytes, which go to your red blood cells. So think of this, in this one life cycle, we have 1, 2, 3, 4 different stages. And this is organisms budding off cells of itself. So it's actually 4 different stages of this organism spreading, and it needs all of those stages to complete its life cycle, and it needs 2 hosts in order to do it. So it needs both the vertebrate with the blood, and it needs the mosquito to transfer it back to the vertebrate, and it's doing some of its reproduction inside the vertebrate, and it's doing some of its reproduction inside of the mosquito. Paramecia is also something we're going to see in lab. And do not memorize this life cycle. It's very complicated. But I just want you to appreciate that it has both haploid and diploid nuclei in the same cell. It has multiple nuclei. And the way it doesn't really make gametes, and what it's going to do is it's actually going to swap, right, its nuclei, and they're going to go meiosis and make new nuclei inside there. And so meiosis is taking a diploid nucleus and going to 4 haploid nucleus. Then it swaps those haploid nuclei among organisms, and then they fuse to make new diploid nuclei, where it already has original diploid nuclei that break down, and then does meiosis and makes more haploid nuclei. Right? And so we refer to these as a micronucleus and a macronucleus, and they're actually separate ploidy levels. Don't memorize this. It's just to give you an idea of how different the life cycles are from humans and animals. Here's one in an algae. This is a brown algae. And it's showing you an alternation of generations, something we'll come back to when I'm talk next time on plants, because they have an alternation of generations. And so you actually see these are two separate living organisms. Here's one algae growing on the rocks, and here's another type of algae growing on the rocks. These ones are much smaller than those. These are separate male and female, and notice they're all haploid. And so the male makes sperm. Notice it has two flagella and one tensile type, so they're very different from the ophistocont, or our flagella. Right? And also it has the stationary egg that gets fertilized. From here, you get the first two end stage called the zygote, and it grows up to the sporophyte plant. So the female gametophyte was down here. And so we actually have different names for that. Phyte means plant. This is a gametophyte making the gametes. This is a sporophyte because it actually be releasing spores. And this is a swimming spore called a zoospore. Right? Do not need to memorize this, but the things you want to notice is, one, it's made up of both haploid and diploid separate plants. Well, actually, they're kelps. We call brown algae kelp. So you have a haploid kelp and a diploid kelp. But also notice, to make more haploid kelp, all the haploid kelp can do is make a diploid kelp. And the diploid kelp makes a haploid kelp. Right? It's not that the haploid kelp are reproducing themselves in the diploid kelp. They're not doing that. So to complete its whole life cycle, it has what we call phases or generations. You have a haploid, a gametophyte generation, will make the sporophyte generation, which will make the gametophyte generation. It sounds silly, but this would be as if in humans, you made your eggs, you made your sperm, and they grew up by themselves. So we're 2n equals 46. Your eggs are n equals 23, or your sperm is n equals 23. And so you'd have, if you were female, your egg would grow up to your daughter, right? And she'd be haploid, 23, and go off by, right? Or male, your sperm would grow up into a son, and he'd be n equals 23 and go off by himself, right? And so during your lifetime, you can make haploid kids, right? Then your haploid kids would have to get together with partners, right, to make your next generation, which would be the diploid grandkids, right? Does not happen that way in animals, obviously, but that gives you just conceptually kind of an idea of what's going on here. The sporophytes are only making the gametophytes, and then the gametophytes get together to make the next spore. Okay, we have two ways that cells divide. And so we have mitosis, and in 130 or cell biology, you learn all the steps and the stages. That's fine for that. We only need to know the main outcome. In mitosis, things you need to know is that it makes identical copies of eukaryote cells. Bacteria, archaea do not do mitosis. They do binary fission, which also makes identical copies of the genophore, right? But it's a different process. So in eukaryotes, we have multiple separate chromosomes, and this is a process for duplicating and separating those chromosomes. So it's called mitosis. One cell goes to two identical daughter cells. So you have one cell will go to two daughter cells, and the original cell is gone. So one cell goes to two. It's not like you have one cell and makes two daughter cells. You have a total of three cells. That cell actually comes and it pinches in and pulls apart. So it literally divides itself into two cells. It can happen in, obviously, we're used to it being in diploid. We think about animals growing. And because sperm and egg in animals do not divide mitosis, don't get confused. I just showed you a life cycle. All of this is haploid. That zoospore grows by mitosis to make all of these cells. That is all mitosis. And so haploid cells definitely can divide by mitosis, and they do. Diploid cells can, and then ones we'll get to later, and then the fungi are triploid, and or N plus N cells, and in plants we'll get to triploid. They all. So mitosis, just think of a cancerous cell that's messed up with its nucleus. Mitosis doesn't care, and that's actually what makes cancer a problem. It just divides and divides and divides, right? So the ploidy level, haploid, diploid, doesn't matter for mitosis. And in this process where it makes a new organism, it's not changing anything with the genetics. It's called asexual reproduction or cloning. So if you take a branch off a tomato plant, put it in water, it sits down roots and it grows. Now you have 2 tomato plants. That was asexual. It's all by mitosis, cloning, and that's how that happened. And it doesn't change the ploidy level at all. It remains the same. And so that's going to be different in meiosis. Meiosis is a reduction division because we're always starting with a diploid cell, 2N, and it will go to 4 haploid cells. And so 1 cell goes to 4. In this process, obviously, if you're starting with 46 chromosomes in us, and your eggs have 23 chromosomes each, they're not passing all the chromosomes along. And there's a sorting mechanism to make sure you get a complete set. In that process, it also can mix up similar genes. And so all of the cells are a little bit different. And this is where your sexual recombination comes from. It's not mutation, it's mixing up genes that are there. And so in your life, you're not going to make 2 identical eggs or sperm. So that's why you can have the 2 same parents. So if we were together in class, I'd ask how many people have a sibling. You raise your hand, right? Are they identical? And I'm like, well, if your sibling is full sibling and you have the same parents, why aren't they the same if you're starting with the same genetic stock? And that's because in meiosis, right, every time your parents were making the eggs or the sperm, it's mixing up the genes. Not mutation, just mixing, recombining. And this is involved in the sexual reproduction phase. Right? It doesn't always result in gametes. Sometimes it results in spores. Right? If we go back to our kelp life cycle, the product off here were spores, not gametes. The gametes, the sperm and the egg, this is already haploid, that's already haploid. Those are actually being produced by mitosis. And in a lot of your plant and algae life cycles, the product of meiosis are spores, and then later the gametophyte makes the gametes by mitosis. There's no change in ploidy level in that case. Okay, so where did mitosis come from? This is more if you've had 130. Otherwise, you don't really have to worry about it. But if you had 130 and you know about mitosis, it might seem a big jump. It's like, how do you get something so complicated? So I'm showing you here, this is binary fission. And we talked about that, where the DNA, the single loop chromosome of the prokaryotes, the bacteria, in this case, duplicates itself. The membrane pulls apart and separates 2 chromosomes. But when we get to mitosis, we have multiple chromosomes. And I just want to show you some variety, because if you jump straight to plants and animals, like you learn in 130, you might think, how could this be related? But if you look at diatoms and some other organisms, mitosis is taking place entirely inside the nuclear envelope, and the chromosomes in the spindle apparatus actually attaches to the nuclear envelope, and it uses that to pull with the spindle apparatus, pull them apart. And the dinoflagellates, they're actually attached, that's the second stage. First stage is dinoflagellates, where they're attached to the nuclear envelope and the microtubules are to the outside, pulling it apart, similar to the binary fission. Then the microtubules move into the inside, and then we lose the nuclear envelope. And so this is in a nutshell. Don't worry about it. It's not for my class. But if you've had 130, I just want to throw this out there so you can think a little bit about, first of all, there's different for the diversity, because in 130 you're going to learn plant and animal mitosis. That's the whole world, seemingly, but it's not. There's a lot of diversity in the eukaryotes. And so just showing you the way here the diatoms and the dinoflagellates divide. the stages are a little bit different, and they're a bit more similar to binary fission. Okay, lastly, I want to talk a little bit about the importance of eukaryotes in food chains and just modes of nutrition and their ecology. And so there are three basic modes of nutrition. And we already thinking of eukaryotes, you kind of already know that you have plants and then you have animals and fungi. And plants are green and they do photosynthesis, and animals and fungi have to eat other things. And so anything that does photosynthesis we call an autotroph. Troph means to feed, and auto is self. So these are the self-feeders. They use sunlight for energy. And we kind of talked about these different categories. We had 4 categories when we were looking at the bacteria. Here we just pretty much have 2 categories. All right, we have the autotroph and the heterotroph. And so these would be photoautotrophs in bacteria and chemoheterotrophs in the bacteria, but we don't have to worry about that in eukaryotes. We don't have quite as much diversity. One thing I wanted to mention, anything that has photosynthesis is an autotroph. Plants and green algae are just one of the clades. And so other ones are the red algae, the brown algae, the golden algae, the diatoms. A lot of them are a brownish red color. And if you look at our ocean water along shore, it has this deep kind of blue-green brown color. And sometimes people think, oh, that's pollution. That's not. That's the diatoms and dinoflagellates in our water that are the base of the food chain, along with cyanobacteria. Those are the major things feeding everything in our waters near shore, and that's what gives it the color. So again, not all algae are green. There are lots of other colors. On land, pretty much everything's green because it's just plants. But in the water, we have lots of photosynthetic organisms. Heterotroph means they eat other organisms. These smaller ones, what we call protozoa and plankton, they're feeding by phagocytosis primarily. Right? There are some absorptive protists, and those are ones that function more similar to a bacteria where they excrete enzymes and break down, and then they absorb the simpler molecules. But a lot of them feed by phagocytosis. And then we have some that do both, something like Euglena. It can be going along and doing photosynthesis in the sun, and if something juicy comes along to eat, it'll take it in by phagocytosis as well. And so we call those mixotrophs, mixing up hetero and autotrophic means of feeding. Okay, in the protist, remember that's everything in that big clade of the 4 supergroups in the eukaryotes, except for plants, animals, and fungi. So things that aren't multicellular on land. That's all the protists. And one class is really important to our shore and our waters, or what we call plankton. Plankton is anything that's small, and they may swim around, they may go up and down on the water column, but they can't swim against the current. So pretty much they drift with the current. So they can be swimming, but they can't grow where they want to go, right? Many, their whole life is that way. A lot of the single-cell algae and smaller ones, and dinoflagellates and diatoms, for example. Some may also be plankton or a lot of the earlier juvenile stages for a lot of the animals. So when we get to the crabzoe and things like that, when we talk about animals, a lot of the young stages in some of the animals in the water are also planktonic. They just float with the water. They can't control. They're not large enough to swim against the current. And so we divide them into 2 types, kind of like auto and hetero, but we call them phyto and zoo. All right. So the phytoplankton are photosynthetic, phyto meaning plant, and so they're the autotrophs. Zooplankton are the heterotrophs. They eat other plankton. We also see some protists on land, especially in wet soil. They also can be incorporated into guts. So what makes a termite able to eat wood is that in its gut it has all these protists that actually break down the wood, and then it eats the remains of the protists. And so they have lots of different habitats as well, but a lot of them are all focused ones on water. And so phytoplankton are really important because they're the ones that feed the entire marine ecosystem. Just like we'll talk about food chains later on, you think of the green grass, and then the insects are eating the grass, and the mouse is eating the insect, and the snake's eating the mouse, and the hawk's eating the snake. That all comes down to how much energy the grass can obtain. And in the aquatic ecosystem, it's how much energy the phytoplankton can harvest. And usually what's limiting them isn't the temperature of the water, amazingly. It's not the sunlight. It's the nutrients in the water. And so we'll talk about that when we get to ecosystems later on in the semester. Sometimes they can grow very fast, and these are called blooms. And I'll show you a picture from a satellite showing you these algal blooms along our coast. One of them we've talked about in lab, and that's the red tide with the dinoflagellates. That would be an algal bloom of a phytoplankton. In nature, the upwelling of the current is what brings the nutrients up. And so that makes our water near shore very nutrient rich, and therefore we have a really high productivity of phytoplankton, meaning we have lots of organisms right on our shore, and the intensity of the amount of life on our shores is much greater than if you go to the east coast and compare the intertidal zones. You see that. Or the amount of fish that we can capture is because of the great amount of phytoplankton, because of the upwelling bringing the nutrients up. So it's all kind of tied together. And we'll come back and talk about nutrients again when we get to ecosystem ecology. As I mentioned before, just now, that it's not tied to the temperature as much. And actually, usually you think chemical reactions, things grow faster with the warmer temperatures. And we do see that on land with the wet area, also being the tropics, being the warmer area, we get tremendous plant growth in the tropics and not as much in the cooler areas. In the ocean, it's kind of the opposite. And so here showing you, this is very far north in the Bering Sea. This is cloud cover over on the upper right-hand side. What you see swirling here are different types of plankton that are just blooming in huge amounts. And this is a satellite image in real color from a camera taken by NASA. So you can see these huge blooms, and think of all organisms that are relying on that food in that aquatic system. And so the producers are eaten by the consumers, and they move up each of these stages up and feed everything from the smaller fish to the larger fish to the marine mammals on up to us. Here showing you what's happening with increase in water temperature associated with climate change. And in the yellow regions, the sea surface temperature increase, right, has happened. And so where it's going up, you can see it in the orange and the brown, and where it's getting cooler, it'd be a little bit in the blue. And why this is important, if you look here in these waters, anything to the negative side means there's a decrease in the amount of chlorophyll being received. And so what I mean by received, so this is a satellite image. They can take a camera and put a special image on it, and so it will record the amount of chlorophyll. And all organisms that do photosynthesis have at least a little bit chlorophyll, even if they're not green. And I'll go over that a little bit in the lab. So basically, chlorophyll is a direct indicator of the abundance of these phytoplankton. And you can see that the warming temperatures is making the phytoplankton scarcer. And if the phytoplankton are getting scarcer, that's going to cause a collapse, hopefully not soon, but could cause a collapse of the whole marine ecosystem. And so even though we're studying these very small little organisms in lab, their significance to the ecology on Earth is tremendous, especially the aquatic ecology. And so getting up here to show you our little chart here. So these organisms through here. So the reason we're spending time in lab to go through all these different groups is because they're really, first of all, it's a diversity of life showing you. I just showed you one little thing on how they divide by mitosis is different in the different groups. And we'll learn all sorts of different things in lab about their different types of cell walls and all sorts of things, the different pigments that they have for photosynthesis. But also they're very important in all sorts of different ecosystems. So we don't want to forget the base of these ecosystems and also the important links between the stages cycling nutrients. And a lot of these are really important in that. Okay, I hope you enjoyed talking about or learning about eukaryote diversity. And we'll be finishing up this section and starting the next one. We'll be starting our diversity of life, moving up to multicellular organisms and start talking about plants.